Okay, so this is a revision of 11th class for you all. Here, this diagram shows mitochondria. If you see the lower diagram, here you can see the two membranes of mitochondria. This is the outer membrane and this is the inner membrane. The outer membrane has no enfoldings, whereas the inner membrane has these enfoldings, which are known as cristae. The outer membrane is permeable and it has less proteins, whereas the inner membrane is selectively permeable, means it allows the useful substance to pass through it and has more proteins. Because of these two membranes, two compartments are formed. One is this inner compartment, another is this outer compartment. Now, this ground substance of mitochondria is known as matrix. These are ribosomes which are present in mitochondria, and this is the circular DNA of mitochondria. Because of the presence of ribosomes and circular DNA, the mitochondria is also known as semi-autonomous organelle. Semi-autonomous organelle because it has its own ribosomes and it has its own DNA. The ribosomes which are present in mitochondria are 70 S types. 70 S types, and these 70 S types ribosomes are also present in prokaryotes or in bacterial cell. Because of the presence of circular DNA and ribosomes, it can synthesize its own protein and it has its own genetic information. So mitochondria is also known as a semi-autonomous organelle. The enfoldings cristae has some particles on them, some structures on them, which are known as F0 or F1 particles, or elementary particles, or oxysomes. So these are known as they have this name. Now if we take the structure of this portion, only this much portion, it will appear like this. We have this outer membrane, this inner membrane which has enfoldings which are known as cristae. The outer membrane, in between the outer membrane and the inner membrane, this space is present. This space is present, known as intermembrane space. This is one F1 particle which is shown here also by this black color. If you see in this F1 particle, how does this in this cristae, how this F1 particle is attached? This is cristae and these are F1 particles. Like this, many F1 particles are attached here and there on this cristae. This is the structure of one such F1 particle. It has base. This is base which is inside the membrane. It is also known as F0 particle. Then we have a small stalk which is F5 or F6 particle, and a circular structure which is F1 particle or head. So this is base, head, stalk and head. This is F0 particle, this is F5, F6 particle, and this is F1 particle. Now here it synthesizes this F1 particle has enzyme ATP synthetase. Now, because of electron transport chain, the concentration of protons increase in the intermembrane space. This is the inter means in between membrane. One membrane is this, another membrane is this, and this is intermembrane. So the amount of protons, they increase in the intermembrane space. Now always the substance which is more at one place, they tend to move towards a place where they are less. So from intermembrane space, these protons, they try to move into the matrix, this matrix. Now there are special sites through which they can move, and those sites are these F1 F0 F1 particles or oxysomes. They can go only through this. So these protons which are at high energy, they start passing through it. When they pass through it, they release energy, and that energy is utilized to combine ADP and inorganic phosphorus with the release of ATP in the presence of enzyme ATP synthetase. And this is how energy is formed in mitochondria. This was just a revision for you of mitochondria so that you can use it for your NEET examination. Now I move on to the next topic of molecular basis. And that next topic of molecular basis is DNA fingerprinting. We come to the next topic, DNA fingerprinting. Yeah. DNA fingerprinting. Now, the use of DNA fingerprinting, first of all, I'll tell you who was the one who discovered this particular technique. This was discovered by Alec Jeffrey, A-L-E-C, Alec Jeffrey, Jeffrey, in the year 1985. In the year 1985, Alec Jeffrey, he discovered this technique of DNA fingerprinting, and this is used to solve the criminal cases. This is used to solve the criminal cases and paternal disputes. And disputes, that means whether this boy is whose son and all that. There was a political dispute also, this paternal dispute, and that was one of the leader of a renowned leader of one of the parties, and he had to face this paternal dispute issue. So that was solved by DNA fingerprinting. Okay, now what happens in this? In our genome, 99.9 base sequences are same in all the human beings. Base sequences are same in all human beings. So 99.9% base sequences are same in all the human beings. So where lies the difference? Where lies the difference? The difference is just in 0.1%. Here lies the difference. Here lies the difference, and this difference is the identification mark, or we can say on the basis of this, the identification of a particular human being takes place. Identification, that means we all are similar, all the human beings are similar in 99.9 base sequence. In all the human beings, 99.9 base sequences are same, and the only difference between every human being is that 0.1% of base sequence, by which one human being differs from another human being. And that 0.1%, because it is different in every human being, that becomes the mark of identification. On the basis of that 0.1% difference, every human being can be identified. Now, when the DNA of human being is separated by, when the DNA is separated by density gradient, density gradient technique. When the DNA is separated by density gradient technique, we get two peaks. Two peaks are formed. Two peaks are formed. And these two peaks are like this. This is bulk peak. This is bulk. And this peak is satellite and forms satellite DNA. This forms the satellite DNA, and this satellite DNA is repetitive DNA, where this satellite DNA is repetitive DNA. That means certain base sequence in this DNA, they are repeated a fixed number of times. That means base sequence, we will not say all the base, certain base sequence are repeated a fixed number of time in different individuals, That means some base sequences are repeated. Like if we have GCTA, it will be repeated. GCTA, a certain, up to a certain number of times, this is repeated. Say, suppose it is repeated up to 1000 times or 100 times, or the same base sequence is repeated 50 times. So it can be repeated to a fixed number of time in different individuals. And these base sequences are repeated. If you see here GCTA, 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 suppose it is repeated 1000 times. So it will become the satellite DNA. Suppose it has been repeated 1000 times. So it depends. Sometimes it gets repeated only 50 times. So this makes the satellite DNA. This makes the satellite DNA. Now this satellite DNA is of two types. Satellite DNA is of two types. The first is the mini satellite. And the second one is microsatellite. Mini satellite and microsatellite. In mini satellite, 15 to 50 base pairs are repeated 1000 times. 15 to 50 base pairs, BPR base pairs, are repeated 1000 times. This makes it mini satellite. Now, in the case of microsatellite, less than 10 base pairs are repeated. Here, in this case, less than 10 base pairs are repeated 10 to 100 times. Less than 10 base pairs are repeated 10 to 100 times, then we call it microsatellite. And if 15 to 50 base pairs are repeated 1000 times, we call it mini satellite. These mini satellites, they are used as VNTRs. VNTRs are variable number of tandem repeats. It is variable number of tandem repeats. And these VNTRs are also used as radio labeled probes. These are also used as radio labeled probes. That means they form probes. Suppose, which we know, in which the base sequence we know, these are radio labeled. Suppose they form this probe, A, G, C, T. This probe, we know that it is A, G, C, T, and this is radio labeled. When this probe will put into a bath where various fragments of DNA are there, so this probe will attach. Suppose this is put in a bath. This is a bath where DNA fragments are there. Many DNA fragments are there. So this probe will attach to which fragment? Which will have complementary bases. That means it will attach to the fragment which has T, C, G, A. It will attach to this. So when we will see this radio labeled hybrid, when it attaches, we say that hybridization has taken place. So when we will see this hybrid form, we will come to know that this fragment, because about this we know that it has AGCT. So that means in the bath, whichever fragment it attaches, it will have TCGA, and we will come to know that this fragment, the base sequence of this fragment is TCGA. So this is how the mini satellites are used as radio labeled probes. Is it clear, children? Is it clear? Yes, beta. Yes, bache, is it clear? Yes, beta. Okay. Fine. Paras and Sukh Sahaj. Others, as usual. Others. Others, beta. Parthivi, fine. Others are also there. There are 22 people in the class. 22. And out of that, I have got only 2 response. Sanjolika is also there. Amit Katoch is there. Aditi, Gunjan, Girish, Karthik, Simran, Vanshika, Navneet, all are there. But I have got just 2 responses. If you actually, why do I ask? I ask you so that you come, so that I come to know that you have understood it. If you have not understood it, I'll explain it again. That's why I ask. At least, some of you answer so that I come to know that this particular topic you have understood and I don't have to repeat it. But if you don't speak, then I feel that you have not understood. Yes. Yes. Is it clear, children? Yes, Amit Katoch, Navneet. No? Shall I repeat? Okay, Vanithi. Yes. Amit Katoch. Arif. Okay, fine. I've got the reply. Mariam also, fine, beta. I have come to know that all of you have understood. Now we come to the next process. The process, actually, we are studying, this was just an introduction of DNA fingerprinting. Now we come on to the process, which what is DNA fingerprinting and how this is done. So I think you have come to know that mini satellites, out of the satellite DNA, the mini satellites which have 50, in which 15 to 50 base pairs are repeated 1,000 times, they are used as VNTRs, and these VNTRs, they form radio labeled probes which we put in the bath which have DNA fragments. So to whichever these probes attach, we come to know the base sequence of that particular fragment. So this is how it is used. Now we come to the process of DNA fingerprinting. So this was the gentleman Alec Jeffrey who gave us such a nice process to deal with all the disputes. Okay. So many disputes are there in the world, so much crime is also there. So whether it is blood, or it is hair, or it is skin, or it is semen, anything can be used by this process to trace the culprit. So we come to the process, and the process, I think I have given you notes of this. Have I given you notes of this, beta? Have you got the notes of this, or shall I send it again? I think I have given you. Check it, you will have. Okay, so I come to the process of DNA fingerprinting. So the first step of the process is source of DNA. See, what do we need to for DNA fingerprinting? First of all, we should have sufficient DNA. So this DNA, source of DNA, the person or the whatever who are investigating that particular crime or whatever happened, so those people, they go there to collect the source of DNA, and the source of DNA can be 1 microgram of tissue, it can be 1 microgram of tissue. Either they collect this, it can be 1 microgram of tissue or 1 lakh cells, or it can be 1 lakh cells. Either of these are taken. 1 microgram tissue or 1 lakh cells, and these can be from which substance? 1 microgram of which tissue, or 1 lakh cells of which tissue? This can be obtained from blood. These can be blood cells. These can be semen. This can be semen. These can be hair cells. These can be skin cells. Hair cells, as I have already told you, it was done in case of Naina Sahani case, who was a journalist and who was burned alive in a tandoor in Delhi. This happened, and nothing was found because she was brutally burnt in the tandoor. No body tissue was found. Later on, around that tandoor, some hair were found. And when those hair were collected, those hair samples, those hair cells, they were sent to Hyderabad. There is an institute, um, a biotechnological institute, where this DNA fingerprinting and all is done. So these were sent to Hyderabad, to that institute, and in that institute, when the hair cell DNA fingerprinting was done, and the DNA fingerprinting of hair cells were matched with the parents, it got matched, and ultimately it was found that the burned person who was burned, the person who was burnt in the tandoor, was Naina Sahani, and the culprit who did it, he was sent to jail. And I think few days back, only few months back, he is released. This year only he was released. See, after so many years, this happened sometimes in 80s it took place, and now he has been released, the person who committed this. So hair cells can, it can be hair cell, it can be skin cell, and it can be blood cells. And the center which is there in Hyderabad, which does this work very nicely, that is Center for Cellular and Molecular Biology. It is a very, it is a center of international repute, and the people go there to for research work, for RA-ship, research associateships, the research work. So much of research work is going, and the funds, they flood into that institute, because the scientists, the people who are the researchers who are working there, they get good funds also. So it's a wonderful institute in Hyderabad, Center for Cellular and Molecular Biology. There the hair cells, hair of Naina Sahani were sent, where it was found out by DNA fingerprinting that these hair are of Naina Sahani because they matched with her parents. So this is how. So either it can be 1 microgram of tissue or 1 lakh cells of blood, 1 lakh cells, it can be blood cells, it can be semen, it can be hair cell or the skin cell. Now what is done? The second step, we have already collected the samples. Okay, we have already collected the samples. Now it may happen that the sample is not sufficient. That means the person who has gone to collect the sample gets very few pieces of hair, or very little skin, or very little semen, or very little blood sample. That means the samples are insufficient in amount to conduct this process of DNA fingerprinting. So something must be done to amplify that sample, to increase the sample in amount. And you, in the biotechnology, you have done a technique which amplifies the DNA. Yes, Sukhsahaj, that's very good. It's PCR. So yes, Amit Katoch, PCR. Yes. So we have got very little sample. Now, by the process of PCR, this DNA amplification is done. So we can go for the second step, and that second step is DNA, if the sample is insufficient, amplification. This is done by polymerase chain reaction, or PCR. In short, this is known as PCR technique. These days, they are doing it for the COVID also. So if the sample is less in amount, if the sample collected is less in quantity, if the sample collected is less in quantity, then this PCR is done. Why? Why? To increase the amount, to make many copies. In less in quantity, PCR makes multiple copies of DNA. So multiple copies of DNA is obtained if the sample collected is insufficient in amount. So this is done by DNA amplification by PCR technique. Now the third step comes. We have collected now from the blood cells or from the semen or from the hair or from the skin, we will remove the DNA. We will take out the DNA. So the third step is extraction of DNA. The DNA is extracted. In the chapter of biotechnology, principles and processes, you have done this technique, extraction of DNA, in which we use certain chemicals by which the wall, the outer cover, the cell wall, if it is plant cell, and the cell membrane, if it is animal cell, it is broken, it is ruptured, and the DNA comes out, and that DNA is then obtained by separating it with chilled ethanol. Chilled ethanol is used to separate that DNA, and we obtain it. So by using various enzymes, this is obtained. That means by using various chemicals, and finally, and chilled ethanol. When I was doing this with your class, with the previous class means that class of Nikhar and all, this your seniors, Riyad and all, Namita and all. So in that class, I showed also, I think you in the test tube, we had taken out the DNA of onion. I think I showed you. I showed you in the test tube. Did I show you, beta? By adding chilled ethanol, I extracted the DNA. First, it was done with mixie and all, adding some detergent or shampoo, and then yes, this Chitra brought her